Hi, physiology students. Welcome back. This is your recorded lecture, which will be in place of our class on Tuesday. We finished kind of a section of chapter 12 talking about brain death. And I'm going to skip over kind of the meninge part. If you're wondering where we are, we're in chapter 12, part C, PowerPoint. And we finished talking about brain death. I'm going to skip over the meninge layers. You should remember all the meninge layers from anatomy, but we're going to kind of move on. We'll kind of start here with what meningitis is. Remember, whenever you see an itis word, that means an inflammation. So meningitis is inflammation of the meninges, which are the layers that protect the brain. And you can see these layers here. It could spread to your central nervous system, which would lead to inflammation of the brain. And we refer to inflammation of the brain as encephalitis. Meningitis can usually be diagnosed by observing microbes in a sample, cerebrospinal fluid sample obtained from a lumbar puncture. And we'll talk about what that is in a little bit. So this brings us to cerebrospinal fluid and what is CSF. It forms a liquid cushion of constant volume around the brain, so it helps to give buoyancy to your brain. It reduces the weight of the brain because it's sort of floating. It helps the brain float so it's not crushed under its own weight. It also protects the brain from any trauma, and the cerebrospinal fluid will nourish the brain, carry chemical signals. It's composed of a watery solution formed from blood plasma, but with less protein and some different ion concentrations. So cerebrospinal fluid is formed by the choroid plexus, which is a cluster of capillaries that hangs from the roof of each ventricle. It's enclosed by pia mater and surrounds a layer of ependymal cells. The CSF will be filtered from the plexus as a constant rate, and the ependymal cells use ion pumps to control the composition of the cerebrospinal fluid. Normal adult CSF volume is about 150 milliliters, and it's replaced every 8 hours, which is kind of interesting. So you can kind of see here, number one, this is showing the formation of CSF. The choroid plexus, which is found in the roof of each ventricle, will produce the cerebrospinal fluid, and then that CSF will flow from the ventricles into the subarachnoid space, which is the space in between the meninge layers. The CSF will then flow through the subarachnoid space and get absorbed through different sinuses via the arachnoid granulations. And this just is showing how the capillary of the choroid plexus forms the CSF, and then it gets filtered through these ependymal cells. So what is hydrocephalus? This is when some sort of blockage is obstructing flow of the cerebrospinal fluid circulation or drainage. So this will increase pressure in the brain. In newborns with hydrocephalus, their skull bones, remember in the embryo, are unfused. There's soft spots or fontanelles. So increased pressure will actually cause the head to enlarge. And hydrocephalus was a big component of the Zika virus. If a pregnant woman were to get the Zika virus, her children were often born with hydrocephaly. But in adults, the rigidity of the skull keeps the pressure within, potentially leading to brain damage. The hydrocephaly can compress the blood vessels and crush soft nervous tissue. The ultimate treatment is just to drain the cerebrospinal fluid that's been built up because of blockage, with a ventricular shunt to the abdominal cavity. And this is what hydrocephalus looks like in a newborn. Again, it can cause an enlarged head because the fluid gets through those soft spots where the skull bones haven't fused together. The blood-brain barrier then also exists in the brain between your astrocyte cells and your capillaries. This helps to maintain a stable environment for the brain. If there is chemical variations in the brain tissue, this could lead to uncontrollable neuron firing, so it wouldn't be able to control the nervous impulses. Substances from the blood must first pass through a continuous endothelium of capillary walls before gaining entry into the neurons. And these are these tight junctions that ensure what substances pass through and what substances do not get through. And this barrier is formed from the feet of astrocytes and smooth muscle-like pericyte cells, which will surround the endothelial cells. So substances can move through these endothelial cells by simple diffusion or specific transport mechanisms that will help move larger ions. So this is just a look at the brain capillaries. Actually, it shows you the difference between most capillaries in the body and the brain capillaries. And you can see how in the brain, there's tight junctions between endothelial cells. So these astrocyte feet wrap themselves around the capillaries to only allow certain things to get out of the blood capillary into the brain interstitial fluid. The blood-brain barrier, the thick basement membrane surrounding the capillaries, is the last part of the barrier. This will contain enzymes that destroy certain chemicals. The blood-brain barrier will be absent in some areas of the brain, like your vomiting center and the hypothalamus. And this is necessary to monitor chemical composition and temperature of the blood. So you want those areas of the brain to always be able to receive the signals that it needs to. Brain injuries and disorders. You should be familiar with some of these words because you might run across them again in your clinical or healthcare related field someday. A concussion is a temporary alteration in function of the brain. A contusion is permanent damage. And a subdural or subarachnoid hemorrhage has to do with pressure from blood may be forcing the brainstem through the foramen magnum, and that could result in death. And then the cerebral edema, the word edema means swelling. So this is just swelling of the brain, usually due to some sort of brain injury. Cerebrovascular accidents are also known as strokes. Ischemia is when the tissue is deprived of blood supply, leading to death of the brain tissue. And that's usually because if you don't have blood supply, you don't have oxygen getting to that part of the brain. If a cerebral artery is blocked due to a blood clot, glutamate will act as an exotoxin, which will actually worsen the condition. Hemiplegia is paralysis on one side, or sensory, where sensory and speech deficits may result. Transient ischemic attacks are temporary episodes of damage that is reversible, and tissue plasminogen activator is the only approved treatment for stroke. Alzheimer's disease, often called AD, this is progressive degenerative disease of the brain that will result in dementia. Key proteins appear to be misfolded, they're malfunctioning. Alzheimer's disease is characterized by memory loss, short attention span, irritability, moodiness. You can read through the others, hallucinations. You get plaques of these specific proteins that are forming in the brain. Neurofibrillary tangles inside the neurons can also interfere with transport mechanisms, which will eventually kill brain neurons. So basically, for some reason or another, whether it's these tangles or plaques of proteins, you're killing off your brain cells, and the brain eventually shrinks. And when different parts of the brain shrinks, depending on where that location that is, Alzheimer's patients will lose these or progress to these symptoms. So this is look at normal, an EEG of a person with normal versus someone with Alzheimer's. And again, Alzheimer's, their brain cells are just dying off in certain areas of the brain. Parkinson's disease is the degeneration of the dopamine releasing neurons. Basal nuclei deprived of dopamine become overactive, resulting in tremors at rest. So Parkinson's, one of the first keys you look at for Parkinson's disease is someone presents with tremors, so kind of uncontrollable shaking of their hands or fingers. The cause is unknown, but theories include mitochondrial abnormalities, so something wrong with the mitochondria, and also their proteins have started degrading. Here are some treatments. There is some research into stem cell transplants that is promising. Michael J. Fox is the big one with Parkinson's disease. Huntington's disease is a hereditary disorder that is fatal. It's caused by the accumulation of the protein huntingtin in the brain cells, which will lead to degeneration of these basal nuclei and the cerebral cortex. Initial symptoms will include wild, jerking, flapping. It's later marked by mental deterioration. And it's usually fatal within 15 years of onset. The interesting thing about Huntington's disease is it is hereditary, but for women or men who get Huntington's disease, they don't usually get it until later in life. So they don't know they carry that gene until later in life. A simple test, so these are some diagnostic procedures for assessing central nervous system. Basically, any sort of test that will test the reflexes, because any abnormal reflex could indicate something is wrong within the brain, whether it's a hemorrhage, multiple sclerosis, hydrocephalus. They can also take CT, MRI, or PET scans to look at specific areas, maybe using radioactive tracers to help visualize these specific areas and where something might be going wrong. Cerebral angiography uses x-rays with dye to pinpoint any stroke-causing clots, and also an ultrasound. Ultrasound can also be used to evaluate blood flow through these arteries feeding the brain. So that is the end of part C, and we're going to finish up part D here all together with, so give me a second here, and I'm recording this, so you'll just have to bear with me as I find the next part, and we'll move on to part D, and then we'll be done with chapter 12. Chapter 13 we'll talk about. Chapter 13 will be about your senses, so maybe a little more interesting there. But here is part D. And again, part D is a little bit shorter, and I wasn't going to include it, but it talks a little bit about the spinal cord and reviewing some of the anatomy of the spinal cord, which will be helpful. So here is the anatomy and protection of your spinal cord. It's enclosed in the vertebral column, and it will end at your L1 or L2. Your spinal cord functions are that it provides two-way communication to and from the brain. And there's also a reflex center in the spinal cord where reflexes are initiated and completed at the spinal cord level. So here's a look at the spinal cord. It ends at the filum terminale, but about the, let's see, probably the L1 segment, we have this cauda equina, which are spinal nerves coming off the base of the spinal cord as well. So your spinal cord is protected by bone, the meninges, cerebrospinal fluid. The epidural space is the cushion of fat and network of veins in the space between the vertebrae and the spinal dura mater. Cerebrospinal fluid will fill the subarachnoid space. And this is around L1 or L2 would be the site of a lumbar puncture or lumbar tap. And this is showing kind of right between L4 and L5 where you might get a lumbar puncture, needing entering into that subarachnoid space to test your cerebrospinal fluid for different levels of proteins or white blood cells. The spinal cord terminates in a cone-shaped structure called the conus medullaris, and some of this might be a little bit review from anatomy. The cauda equina are all the collection of nerve roots at the inferior end of the vertebral canal. And the spinal nerves are part of your peripheral nervous system. They come off of the spinal cord by 31 paired roots, so 2 on each side. And each spinal cord segment is designated by the paired spinal nerves that arise from it. So here's again, you can see the spinal cord coming down. You also see kind of slightly the spinal nerves coming off. This is a kind of a better picture maybe of the spinal cord coming down. And you'll see the spinal nerve rootlets coming off of the spinal cord at different levels. And some more looks here showing the cauda equina at the base of the spinal cord at the end of it when the spinal cord ends. Here it looks at the curvature of the spinal cord. It follows the curvature of the vertebra as well. If we look at cross sections, there's 2 lengthwise grooves that run the length of the cord and divide the cord into right and left halves, whether it's on the anterior or posterior dorsal side. Gray matter is located in the core and white matter is on the outside, and the central canal will run the length through the cord filled with cerebrospinal fluid. So this is hopefully a review from anatomy. You can see the gray matter, butterfly shape, white matter surrounds it. You can see kind of the spaces labeled here. The spinal meninges would be very similar to the meninges found surrounding the brain. Here is the dorsal root ganglia leading to the dorsal root, and the ventral root will come out the front. And again, whenever you see the word ganglia, that just means a cluster of neuron cell bodies. So here, showing the different layers surrounding the spinal cord, the different meningeal layers. Here's the dorsal and ventral horn, and then there's also a lateral horn in the gray matter. And again, dorsal means posterior, so the dorsal horn will be connected to the dorsal root and the dorsal root ganglia, and the ventral root will be connected to the front side of the spinal cord. Gray matter and spinal roots. So here's the cross-section of the cord, resembles the butterfly or the letter H. And then we have the dorsal horns, ventral horns, and lateral horns. They will all contain different types of neurons. The dorsal horns, again, these are the ones in the back. These are the interneurons that receive all somatic and visceral sensory input from the dorsal root of the spinal nerve. Ventral horns will contain some interneurons, but mostly the somatic motor neurons, which will initiate the response out of the ventral or front roots. And the lateral horns are only in the thoracic and superior lumbar regions, and these will contain sympathetic neurons. So this describes the gray commissure. This is the bridge of gray matter that connects the masses of gray matter on either side. This will enclose the central canal. Your ventral roots exit the spinal cord from the ventral side, so the front side, and the dorsal roots include sensory input to the cord. Dorsal root ganglia are cell bodies of sensory neurons, and spinal nerves are formed by the fusion of dorsal and ventral roots. The gray matter is divided into 4 groups based on the somatic or visceral information. So we abbreviate these: somatic sensory, visceral sensory, depending if the sensory information is coming from your visceral organs or your muscles. And then we have motor output, visceral motor output, or somatic motor output. And these are the locations. So what happens here is any sort of sensory input in blue and green, whether it's somatic sensory or visceral sensory, all sensory input goes through the dorsal root ganglia, through the dorsal root or the sensory root into the dorsal horn, where there'll be interneurons, where the sensory information will synapse. From there, the sensory information will travel up to the brain, or a reflex will shoot out some sort of motor response. And all motor responses go out of the ventral root or the front root out of the spinal cord. So in yellow and red here, you can see the visceral motor, which would go to any sort of motor response to your viscera, your organs. And the somatic motor neurons will take all the motor effectors to your skeletal muscles. So again, sensory input comes into the back, the dorsal side, and then motor information goes out the ventral root. And you can see these different locations of the sensory or motor neurons and where they're located. White matter runs in 3 directions. So again, white matter is white because there's usually some myelinated nerve fibers. This white matter will allow for communication between parts of the spinal cord and brain. And the white matter runs in 3 directions: ascending, descending, or transverse. So ascending up to the higher centers, descending from the brain to the lower cord levels, or transverse from one side to the other. White matter is divided into columns, and we call these columns funiculi, whether they're on the dorsal posterior side, the lateral, or the ventral anterior side. And each spinal tract is composed of axons with similar destinations and functions. So here you can see the dorsal funiculus, ventral funiculus, and lateral funiculus. And these are just areas in white matter that will kind of be passageways for spinal nerves to run, or neurons to either ascend or descend to and from the brain. We specifically have all these tracts, we call them labeled, and a tract is just a passageway for these neurons to run. So usually ascending tracts are all located in the dorsal white column. So ascending tracts, and again, ascending tracts would take information to the brain, and descending tracts would bring information from the brain back down to the spinal cord level it needs to get at. So ascending tracts are all in blue, descending tracts are all in red. So that kind of gives you where these tracts are located. Spinal cord trauma, so localized injury to the spinal cord or its spinal roots, will lead to functional losses. Paresthesis is caused by damage to the dorsal roots or the sensory tracts, and this will lead to sensory function loss, so the inability to sense either externally or internally what's going on with your body. Paralysis is caused by damage to the ventral roots or ventral horn cells. So paralysis is associated with motor function loss, so you're unable to move a body part or control some bodily function. There's two types of paralysis, flaccid or spastic. Flaccid paralysis is severe damage to the ventral root or ventral horn cells, so your impulses do not reach the muscles. There's no control, so your muscles will atrophy or waste away. Spastic paralysis is damage to upper motor neurons of the primary motor cortex, so the spinal neurons remain intact. So muscles could still be stimulated by reflex activity because your reflex activity doesn't involve the higher motor neurons in the brain, but there's no voluntary control of muscles. So there's no control from your brain telling the muscles when to move. Muscles will often shorten permanently. And then spinal cord trauma, more about this. Transection or cross-sectioning of the spinal cord at any level results in total motor and sensory loss in regions below that cut. So paraplegia is a transection or a cut through between T1 and L1. Quadriplegia is a transection in the cervical region. And this is where you might remember the words paraplegic or quadriplegic. The spinal shock is transient period of functional loss, caudal to the lesion. So if someone has a transection or a cut completely severing some region between T1 and L1, they will lose ability to move their legs. Quadriplegic, they'll lose the ability to move all four limbs because the transection was in the cervical region. And in that region, that's where the nerves come out that control all your upper limbs. Poliomyelitis is destruction of the ventral horn motor neurons by the poliovirus, where your muscles will atrophy. Death may occur from paralysis of the respiratory muscles, especially your diaphragm, or of cardiac arrest. Survivors of polio often develop post-polio syndrome many years later because they've lost so many neurons that they can't get back. ALS is also called Lou Gehrig's disease, destruction of the ventral horn motor neurons and fibers of the tract called the pyramidal tract. So these patients lose the ability to speak, swallow, and breathe, and death will typically occur within 5 years. It can be caused from environmental factors or genetic mutations during RNA processing. All right, and then a couple 4 key points about spinal tracts and pathways. So we're going to talk a little bit about these pathways of spinal neurons, and how they go to and from the brain. They're all part of multi-neuron pathways, and a couple key points to remember is decussation. It means the crossing over of these pathways. And most pathways will cross from one side of your central nervous system to the other at some point. So that's what decussate means, or decussation. And we've learned a little bit already how the right side of your body controls the right side of your brain controls the left side of your body. And that's because these pathways decussate or cross over. A relay consists of a chain of 2 or 3 neurons. Somatotopy is the precise spatial relationship in the central nervous system, will correspond to the spatial relationship in the body. And symmetry refers to that these pathways are always paired symmetrically via right and left. So in order to conduct sensory pathways upward through a chain, this consists of 3 neurons. First order neurons will conduct impulses from any sort of cutaneous and proprioceptors. They will branch diffusely as they enter the spinal cord or the medulla oblongata, and first order neurons will synapse with a second order neuron. A second order neuron is most often an interneuron, the cell body in the dorsal horn of the spinal cord or a medullary nuclei. The axons will then extend to the thalamus or cerebellum of the brain. And then a third order neuron, also an interneuron. These will include cell bodies in your thalamus. Remember, everything eventually gets to the thalamus because your thalamus takes everything and tells it where to go to the brain. From a third order neuron, its axon will extend to the somatosensory cortex, and there will be no third order neurons in the cerebellum. So somatosensory signals, so these are sensory information from your muscles. They'll travel along three main pathways on each side of the spinal cord. Two pathways will transmit somatosensory information to the sensory cortex by going through the thalamus first. The dorsal column medial lamensal pathway, some often abbreviated DCML, and also the spinothalamic pathway. These two pathways provide for discriminatory touch and conscious proprioception. There's a third pathway, the spinocerebellar tracts, which will terminate in the cerebellum itself. So this takes you through the DCML pathway and the spinothalamic pathway, telling you what sort of things it's transmitting. And then the spinocerebellar tracts are ventral and dorsal tracts that are used to coordinate muscle activity, so they'll convey information about muscle or tendon stretch. So this is just showing the spinocerebral pathway in blue and the DCML pathway in purple. And it's showing how one often takes the proprioceptors, so coming senses from your muscle spindles. One will take touch receptor sensations, and they'll follow a specific pathway up to the brain. You'll see here that in purple, they will decussate or cross over in the medulla oblongata for the DCML pathway. And you can see here how the spinocerebral pathway will stay on one side of the body all the way up. So again, just showing how different tracks bringing different sensory information to the brain. Some will decussate, crossover, some will not, but the DCML pathway specifically decussates in the medulla oblongata. That might be a test question because this is a very important pathway, and it's important because it'll affect what side of your body is sending out sensations to which side of the brain. And then this is the spinothalamic pathway. It will decussate, you can see here at the spinal cord level. So once a temperature receptor enters into the spinal cord, it crosses over at that same level, and then it travels up to the brain from there. So this is a look at the spinal cord tracts, their origin, termination, and function. It should also tell where it will decussate or cross over, which is important to know. I'm going to ask you to know these three kind of pathways, not necessarily understanding the difference between lateral and ventral for the spinothalamic, but knowing the difference between the DCML pathway, the spinothalamic pathway, and the spinocerebellar pathway, because those are the three main ascending pathways. The descending pathways are either direct or indirect. Direct pathways, we call them pyramidal tracts. Indirect is everything else. Motor pathways involve upper motor neurons, which will be cells in the primary motor cortex, because remember, we're descending back down from the brain, so these will be all motor responses. And lower motor neurons will be in the ventral horn motor neurons, and they will innervate skeletal muscles. So the direct or pyramidal pathways are impulses from these pyramidal neurons in the precentral gyrus. They'll pass through pyramidal tracts. They'll descend directly without synapsing until the axon reaches the end of the tract in the spinal cord. In the spinal cord, the axons will synapse with interneurons or ventral horn motor neurons. The direct pathway will regulate fast and fine and skilled movements. So this is showing a pyramidal or direct pathways. Again, this is a descending pathway to skeletal muscles. Indirect pathways are also referred to as multineuronal. They're complex, they're multisynaptic, meaning there will be many neurons and synapses involved. These pathways regulate axial muscles, so maintaining balance, posture, muscles controlling coarse limb movements, your head, neck, and eye movements that follow objects in the visual field. These indirect pathways consist of four major pathways that are seen here. I will not ask you guys to know these differences. This shows you just in general how these indirect pathways will descend through the rubrospinal tract. And again, just knowing the difference between what a direct pathway is in general, again, there's two types of them, and the indirect pathway in general, but you don't need to know each specific indirect pathway. This is just showing a really cool view of where these pathways would go or originate. This is a diffusion tensor magnetic resonating image that reveals fiber tracts in the brain and spinal cord, which is just a fascinating picture. It's nice to see this in real life, to know that no one just made this up. These are actually the tracts and fibers of the neurons traveling to and from your brain. So kind of cool to see there. And that's the end of chapter 12. So this will be your PowerPoint for Tuesday night that I'll be gone for. We will start chapter 13 and spend the next 2 class periods on chapter 13, which will be on your senses. Thanks for listening, guys, and I hope you're doing well.